I am Mrs. Sloan, and this is video 3 in our genetics series, and this is intended for my AP biology students. So we have already gone over an introduction to Mendel. We have gone over his principles. We have done monohybrid and dihybrid crosses. We've reviewed chi-square tests, the product rule, the summative rule. So if you need help with any of those things, I encourage you to watch an earlier video. In this particular series, in this video, what we're going to do is we're going to go beyond Mendel's principles, and we're going to look at some exceptions to his rules. So first thing we're going to do is review those three. By the way, my students use the notes that are down in the descriptor of the video. Column 1 is a scaffolding, and I will help you fill in your notes. And column 2, I encourage my students to add in pictures or images that are helpful. Okay, so we have the law of dominance, the law of segregation, the law of independent assortment. And I just want to review those three before we go on. Here, you're looking at a dihybrid cross, the classic poster child cross of round versus wrinkled and yellow versus green peas. So the law of dominance would tell you the trait round, or the allele round, is dominant over the wrinkled trait, and that yellow peas are dominant over green peas. That would be what the law of dominance would say. The law of segregation says in your diploid organism, when you form your gametes, you're going to have the law of segregation basically says you're doing anaphase 1. You are separating the homologous pairs, then you will separate the sister chromatids in meiosis 2. So you'll separate the Ys from each other. No gamete will receive 2 Ys, and you will separate the Rs from each other. That's the law of segregation. The law of independent assortment would say you could have a big Y with a big R, or a big Y with a little r, because those are on different homologous pairs and they can assort independently of each other. So now our goal today is to look at exceptions to that. When does that not work out? And it's normal, right? Okay, so the first thing I want to talk to you about are blood types. And when you look at blood types, hold still there. When you look at blood types, we know there's A, B, and O. So there's 4 blood types, and then you have a different factor, the Rh factor, so it could be positive and negative. So how do you get 4 different phenotypes? Well, because you have more than 2 alleles, this is multiple alleles. And so in multiple alleles, you have big IA, big IB, and little I. So big I's are still dominant over little i's, right? So how do you get type AB, and O blood? So any combination of a big IA with another big IA or a little I, that's how you get type A blood. So you can either be homozygous for A or heterozygous for A. I need to get a pointer. Hang on one second. Okay, for type B blood, same thing, homo or heterozygous. There is only one way to be AB blood. You need a big I allele, big IA, and a big IB. Now, here's another exception to the rule. Because you have multiple alleles, you have multiple phenotypes as a result. You just don't have those 2. And also, you have an example of codominance, because both the A allele and the B allele are dominant. So that means that red blood cell has A antigens on it and it has B antigens on it. And then type O blood is homozygous recessive. Okay, homozygous recessive. So another way of writing that, let me move myself over here. Another way of writing that is like this. Okay, and what I would like you to do is I would like you to cross a heterozygote A with a heterozygote B, and then tell me the genotypes and the phenotypes of the children. Okay, heterozygote A with a heterozygote B. So you might want to pause the video and either work it out digitally or on a piece of paper. If you're in one of my students, then on the Ed puzzle, I'll be asking a question. So if you want a little bit of help, a heterozygote A would be crossing an individual like this, with a heterozygote B would be an individual like this. All right, so hopefully you would set up the cross to look like this, right? Big IA, little I, big IB, little I. So how would that turn out? How would that turn out? So you would expect, as far as your zygotes would turn out, you would expect one to be IA, IB, one to be type B blood heterozygote, one to be type A heterozygote, and one to be O. So in this case, your genotype and phenotype ratios are going to be the same, right? 1 to 1 to 1 to 1. So on your notes, if this is 11.4 in my notes, multiple allelic traits, these are defined as when a gene has many allelic forms or alternative expressions instead of just two choices, as in big A and little a. Okay, you could have big IA, big IB, or little I, so any combination of these three alleles to get your different genotypes and phenotypes, right? Codominance is when both dominant allele possibilities are expressed, as in AB blood, okay? When both dominant allele possibilities are expressed. All right, it's one exception, right? Because normally the exception to the rule here is the law of dominance, right? Because usually we'd say, you know, big A is dominant over little a, right? But this is an exception because both alleles are expressed, both big IA and big IB. So that's an exception to the law of dominance. All right, there are some practice problems here my students will do in class. I encourage you to try, and if you need these, I will give you the answers down in the descriptor of the video. All right, so let's look at another exception to the law of dominance, and this is what's called incomplete dominance. And I just am having trouble moving there. Stay there. Okay, so in incomplete dominance, to our eyes, this looks like blending. So you could have a red flower, homozygous R1R1, with a white flower, homozygous R2R2, but in incomplete dominance, so not codominance, but incomplete dominance, it appears to be a blending of the two. Now, truth be told, incomplete dominance could be a factor of codominance. For instance, you could be fully expressing the red and fully expressing the white pigment, and so to our eyes, it appears pink, right? But when it looks like blending, that's when they tend to call it incomplete dominance, when it looks like in between the two. So what happens when you cross two hybrids? Well, you're going to get the red to show up, and you're going to get the pink to show up, and the white. So this genotype and phenotype are going to match, because the genotype is 1 to 2 to 1, and so too is the phenotype. And that's when you know you're dealing with incomplete dominance, when the genotype and the phenotype are exactly the same, because each genotype has its own phenotype, right? So on your notes for incomplete dominance, when a heterozygote has an intermediate phenotype, between that of either homozygote. So one example in plants is red flower crossed with white flowers, and you get pink flowers. So each genotype has its own phenotype. All right. Now, I encourage you to try this. This also works with hair. So straight hair is H1. I can always remember that because the number 1 is straight, right? And curly haired is H2, because 2s are curly. So a wavy, if you had these two mating, then you would have an H1, hybrid, and that would be wavy hair. So then I'd encourage you to do this as practice, to cross two wavy-haired individuals, and you'll see that the straight hair genotype and phenotype will show up, as well as the curly again. All right, so could two wavy people have a straight hair child? Yes. Could you can kind of look at all the different possibilities with all those different crosses if you wanted to. All right, now incomplete penetrance. Penetrance means, like, how likely is this to be expressed? So you could have the genotype for something, the genotype could be present, but you don't actually see it in the phenotype. So on your notes, the dominant trait is not always fully expressed. So you could have, in this family, the pink is genotype and phenotype are present. So here in the pink, he has both the genotype and the phenotype, and the yellow is the genotype and phenotype absent. If you look at this son that they had in green, the genotype is there, but the phenotype is hidden, even though it's a dominant trait. So you could have a trait that has, let's say, 80% penetrance. So even though you got the dominant gene, it's only expressed 80% of the time. And an example in humans with this is polydactyly. And polydactyly is when you have multiple digits. And I'll give you a picture of that in just 1 minute. So I wanted to show you these terms. Complete penetrance, you get it for sure. Incomplete penetrance, some individuals do not express the phenotype even though they inherit the alleles. And I said polydacty is an example of that. And then variable expression is expressivity. It's how you express it. So for instance, you could have the gene for polydactyly where you have multiple digits, but how many digits will you have? That's variable expression and expressivity. So for instance, you could have polydacty where you just have one extra digit, or you could have multiple digits as a result of that. All right, next, pleiotropy. Pleiotropy, what that is saying is, okay, what that is saying, wow, okay, I'll try to move myself. Lots of struggles tonight. Okay, you have a single gene, okay, but there are multiple traits that come along with that. So for instance, in a previous video, we talked about Down syndrome, right? So there's just not one thing, one characteristic with Down syndrome. There are multiple characteristics. So that's what pleiotropy means. So pleiotrophic effects, a single mutant gene affects two or more distinct and seemingly unrelated traits. Two or more distinct and seemingly unrelated traits. And just in case you're wondering, this is not required for the AP exam. So an example in humans is sickle cell disease. So this is one abnormal gene, but yet it causes all of these different characteristics: physical weakness, anemia, heart failure, pain and fever, brain damage. All of these characteristics come from this one single mutated gene. All right, next I want to talk about phenotypic plasticity. So remember, phenotype is how something looks, right? The appearance of it. So in this case, it's the ability of one genotype to produce more than one phenotype depending on the environment. So if you have no plasticity, okay, like you see in this first graph, no plasticity, it doesn't matter what your environment's going on, you're going to get the exact same trait. If you have plasticity here, depending on how this environment changes, determines how the trait will be expressed. So this one would be a highly variable in its plasticity. So the ability of one genotype to produce more than one phenotype when exposed to different environments. And I want to give you several examples of that. And all of this boils down to nature versus nurture. Nature versus nurture, like how much of it is in your DNA and how much of it is because of how you're nurtured, the environment that you are in. So as far as height goes, boy, this has happened the whole time. I'm really sorry there. So as far as height goes, could a person's height, that phenotype, be impacted by the environment? So this is a bell-shaped curve showing height. The dark blue are biological males. The white colored shirts are biological females. How much of that is determined by our genotype and how much of it is by our environment? Maybe if you get good nutrition or not. Another example, here Jennifer and Karen, they are identical twins, and they were born with genes that absorb fats twice as fast as the average person. So they have the genes to possibly be obese. Jennifer has chosen to eat healthy foods, low in fat, and Karen, her twin, is eating a lot of, this looks very McDonald's-y, eating a lot of fatty, unhealthy foods. Jennifer, her obesity genes are not triggered, but Karen's obesity genes are triggered. So depending on the food eaten, so the environment that you're in will determine on whether or not that gene is expressed. So if it's above the genes themselves, if there's some switch that is going to be turned on, some protein that has to be present. Remember when we talked about heterochromatin versus euchromatin, heterochromatin is tightly coiled DNA and it's not being expressed. Euchromatin is expressed. So if there's something in your environment that causes that DNA to unwind, to go from being heterochromatin to euchromatin, that would be referred to as epigenetic inheritance, something above the DNA. So height or body composition has a lot to do with nutrition. So that's what you want to add on your notes, nutrition. Okay, another one is PKU. So PKU, the issue there is if you have excess phenylalanine, and you can't get rid of it. And that phenylalanine is an amino acid. It's right here is phenylalanine. Okay. And if the phenylalanine accumulates, it can cause rashes in babies. I'm saying babies because you will know early on, causing rashes, seizures, hyperactivity, and mental retardation. So as soon as a baby is born, they're tested to see if they have the ability to break down phenylalanine. And if they don't, then what you need to do is, because you don't want to have excess phenylalanine because you don't have the enzyme to break that down. So it's managed by avoiding foods high in proteins containing phenylalanine, and then that's never triggered. So on your notes, PKU, people who are homozygous cannot break down the amino acid phenylalanine, and it builds up to toxic levels in their bodies. It keeps the brain from developing normally. It's leads to intellectual disabilities, seizures, and mood disorders. The treatment is to give affected babies and children a diet low in phenylalanine. So then that enzyme that you need, you're not going to have the ensuing traits because you're not going to have the buildup of the phenylalanine. Okay, next one is temperature, how temperature can affect your environment. So these are Himalayan rabbits. This is how they normally look right here in this top picture. Their ears are a little bit darker, right in front of their face are darker, and their feet. And what scientists have done, though cruel as it sounds, is they shaved the backs of the Himalayan rabbit and put an ice pack on there. I don't know for how long, 48 hours, I don't know, 3 days, a week. And when they removed the ice pack, when the hair, because they had shaved the back, when the hair grew back, it grew back black. And I want you to think about that as an adaptation, because you know this, right? When you see the color black, like my shirt, what light is that absorbing in this room? It's absorbing all the light and it's not reflecting any light back to you. So that would help me stay warmer. If you had a white shirt on, right, you're reflecting all the light. So think about that. The ears, they're very thin. They could get very cold. They are black, so they are absorbing more heat. Where you're breathing in via your mouth, bringing cold air towards your face, that is black. So that area absorbs more heat. And where the feet are, walking on cold snow, that makes sense, right? That's an adaptation that was probably selected for in order to protect the Himalayan rabbit. So wherever the body is extremely cold, that hair grows in black. Now, there's also examples of what sex you are. So whether it is temperature determined, temperature, sex determination, or genotype sex determination. So for humans, it's genotype, right? But in some reptiles, amphibians, and fish, it's the temperature that the egg is kept at. The embryo, when the embryo is developing, determines whether it will be male or female. So on temperature, I would like you to write on your notes, temperature-dependent sex determination, TSD in some reptiles, amphibians, and fish, as opposed to genotypic determination, GSD. In TSD, environmental temperatures during a critical period of embryonic development determines whether the egg develops into a male or a female. So all of these things I've given you, right, this is determining how your gene is expressed. So it's not just if it's dominant or recessive, right? It's your environment influencing your expression. Okay, where polyphenism, okay, sorry about that. Polyphenism, what you eat determines how you will develop, whether you will be a worker bee or whether you will be a queen, depending on what you are fed. So polyphenism, on your notes, the phenomenon where two or more distinct phenotypes are produced by the same genotype, major reason for the success of insects. And why is that? It allows them to partition life history stages. So with larvae, they can be dedicated to feeding on growth, and adults are dedicated to reproduction and dispersal. So depending on what they eat, will determine what they are focused on metabolically. Are they focused on growing, or are they focused on reproducing? Also, to adopt different phenotypes that best suit predictable environmental changes, seasonal morphs to cope with temporarily heterogeneous environments. So do we stay here or do we disperse? And to partition labor within the social groups. So are you going to be a queen or are you going to be a worker? So in this case, this is help guiding them depending on their environment, depending on their life stages, and depending on what is needed in their population. All right. And so this would be an exception to the rule, right? Because it's not what your genotype is, what trait you have. It's what you have in your environment, and what you are exposed to determines how that gene is expressed. All right. And now polygenic inheritance. Now, don't confuse this with multiple alleles. Polygenic inheritance, polygenes, many genes. So it's not just one pair of alleles that determines your trait. It's multiple pairs of alleles. So in this example, as far as skin color goes, it's not like you're either black or you're white, right? There are all degrees of color that you might be, and it's a combination of many different genes. Polygenic, multiple genes, right? So to be very dark, you would be homozygous dominant. To be very light, you would be homozygous recessive. Now, how is this different from multiple alleles? Remember, multiple alleles would say that there's variations on the A, right? You could have big I A, right? You could have variations on the I. You could have big IA, big IB, or little I. So variations with one pair of alleles. That is multiple alleles, right? It's just not big A and little a, or big I and little I. Polygenic is multiple genes. Let me give you another example. Your intelligence. It's not like big S, you're super smart. Big S, you're semi-stupid, and little s, you're super stupid. It's not like that. Your intelligence is a polygenic trait. Multiple different genes are contributing to your intelligence. So polygenic defined as when one trait is governed by two or more sets of alleles. When one trait is governed by two or more sets of alleles. Dominant alleles have a quantitative effect on the phenotype. It adds to it. The more genes involved, the more the bell-shaped curve. An example would be skin color, but this trait is also influenced by the environment, right? What you're exposed to. Next on that is, here is that bell-shaped curve, right? Okay, and the next one, okay, is on wheat and colorations of wheat. And then polygenic disorders. So these are called multifactorial traits. So traits controlled by polygenes are subject to environmental influences, right? Just like skin color is susceptible to environmental influences. So example would be cleft lip, hypertension, diabetes, schizophrenia, allergies, and cancers. All of those are polygenic, so they're highly influenced by the environment that you are in. Okay, next one. All right, is epistasis. Epistasis means to stand upon. So for instance, you could have the alleles for blonde hair and you could have the alleles for red hair or brown hair, but first of all, you have to have the alleles for hair. So if you have the gene for baldness, it doesn't matter what color your hair is going to be because you're not having any. You first need that epistatic gene to give you the green light, and then how it is expressed will be a second pair of alleles. So for instance, if in dogs, okay, you could have, if you look here, the epistatic gene is this capital E. So as long as you have one big E, then you're going to have colored fur. If it is a big B anything, you will have black fur. If it's homozygous recessive, you will have brown fur. And if you do, it doesn't matter if it's big B or little b, if you are homozygous recessive for the epistatic gene, then you won't have that coloring. You would be like, say, a blonde coat. So epistasis, again, not required for AP. A gene at one locus alters the effects of a gene at another locus and may not let it be expressed. So for instance, you could have the pigment. Remember when we looked at the polygenic trait of skin color? You could have a pigment for skin color that you're going to be medium color, but if you're an albino, it's not getting expressed at all. All right, that would be the epistatic gene. All right, so the next exception we want to talk about is human sex determination. So for us, right, XX is female and XY is male. So what I want you to see right away is that girls have 2 copies of the X chromosome, whereas boys only have 1 copy. Again, back to the law of dominance, right? The law of dominance is going to say that you have to have 2 recessive alleles for you to get a recessive trait. Not so for men, not so for boys, because they only have 1 copy of their X allele. And it doesn't matter if it's recessive, you're going to get that trait because that's the only allele that you have. So first, let's talk about sex determination and that gene as well. So if we did a Punnett square and you had a female mating with a male, right? So you would expect half of them to be girls and half of them to be boys. Where you might have an exception to that is it's not really so much the Y chromosome as it is the Siri gene. So the Siri gene is on the Y chromosome normally, but if you somehow do a crossing over event and the X chromosome gets the Siri gene, then even though you're XX, you will be male. And over here, if you are missing the Siri gene on your Y chromosome, if somehow you get a deletion, then even though you are XY, so your chromosomes would look and appear that you should be a male and you would have no bar bodies, right? But you are going to be a female because you're missing the Siri gene. So the Siri gene is the sex determining region Y gene on the Y chromosome that controls development, And then X-linked genes are genes that have nothing to do with gender, yet are carried on the X chromosome. So we're going to talk about that X chromosome. And the person who did the research on that is Thomas Hunt Morgan. And what he worked with is he worked with fruit flies. And fruit flies only have 4 homologous pairs of chromosomes, okay? 1, 2, 3, and 4. This 4th one is really tiny. And if they are XY, they are male. And if they are XX, then they are female. Okay, so he started crossing mutant fruit flies, and maybe the male had the mutation, maybe the female had the mutation, and from that he learned about the inheritance on the X chromosome and the patterns that you would expect. So the first thing I want to show you is just quick difference between males and females. So you're going to see you have striping on both females and males. The females has a few more stripes. The male has that heavier dark pattern right here, that pigmentation. And then also, if you look, let me show you back here on the female right here, how she has more of a point on the end right here, and he has more of a rounded end. You can see that here a little bit, the point versus that rounded end. But where this becomes important is when we start following traits and when you do Punnett squares. So some traits are found only on the X chromosome, and since males only have 1 X, then they are subject to whatever they get on that 1 X. Whereas females have 2 X's, then we have a chance, if we have a bad X, we have a chance that the good X is going to compensate for that bad X. So for us females to get a recessive trait that is found on the X chromosome, we have to have 2 bad X's. So here, let's say, I'm talking fruit flies now. If you crossed a female red fly with a male white-eyed fly, then you would, let's say it was a homozygous red, which I'm now showing you, you write the allele up on the X chromosome. So if you're homozygous red, you're going to be XR, XR. If you are a male, to have white eyes, you have to have the recessive trait, which would be a little r. So let's cross these 2 flies together and see what we get. Okay, so I did the Punnett square for you. Here's the female, here's the male. Look how I wrote in that Punnett square. So this is a carrier female, right? Because she has an X big R, she's carrying the mutation for white eyes. That's not normal, that's a mutant. Okay, so she doesn't have white eyes, she's just carrying it. And you have that same situation right here. This male right here, all the males are going to end up having the red-eyed trait because they get one kind of R, and the female was homozygous red. So all of the males are going to receive that, right? Because the only thing they're receiving they'll never receive the white, because in order to make them male, they have to receive the Y chromosome. So when you do a cross like this, where the female is red-eyed, that's normal, okay, which is called the wild type eye, and you cross it with a mutant white-eyed, and all the offspring are red, okay, that is a good example there of a sex-linked trait. Now, let's cross. Let me move myself over here. Let's now cross these F1s with each other, and so we will produce F2s. So the females are all going to be heterozygotes, right? X big R, X little r. Notice we're not putting any alleles on the Y chromosome, none. Okay, now there are alleles on there. We're just not following those. Right now we're following the ones on the X chromosome. And so we put the male over here, X big R and Y, because that's the only kind of genotype we have for the males, and that's the only genotype for the females. I would encourage you right now to pause me and do this Punnett square and look at the results you have and do your genotype and phenotype ratios. And by the way, I haven't given you any more notes on that because I completed all the notes for you from here on out. All the notes are already done. Okay, so if you want these notes, just get them from the descriptor. So do that cross. Did you do it? All right. So this would be how that cross turns out. So this would be a red female, should be homozygous red. This would be a carrier red female. And here we have the red male, and here we would have a white male. So think about this. How would you get a white-eyed female? I'm riddling, probably going to ask you this in the Ed puzzle in just a second. How would you get a white-eyed female? She would have to be X little r, X little r. What does that tell you about her parents? Would the mother have to be white-eyed, or could she be red-eyed? Would the father have to be white-eyed? Okay, so think about it. To get two little r's, right? One of those little r's has got to come from dad, has to come from dad. So the dad would have to have white eyes. The mother could have white eyes, or she could be this, a carrier, so that the offspring, a female, right? A female to get a sex-linked trait, her dad has to have it because he has to give her one of the bad copies of the allele. That's the pattern that you're going to see. So here we go. So that cross we just did, if you need to look over it one more time, you can. Here is just a picture, an image of that, and the genotypes and the phenotypes. But let's look at this Punnett square right here. Now, how are you going to, Punnett square, sorry, this pedigree. How are you going to be able to tell pedigrees? In a previous video, I already taught you the patterns you would look at for a dominant trait and a recessive trait. Now, what's the patterns you would look at for a sex-linked trait? Well, the sex-linked traits you would get in my class and anything on the AP exam, the sex-linked traits are always going to be recessive. Sex-linked dominant traits are a little bit more difficult to differentiate out in a pedigree. Okay, so just assume any sex-linked trait you're going to get is going to be recessive. So first of all, what you know with recessive traits is they can skip generations, right? Dominant traits cannot skip generations, but recessive traits can. So first thing you would look for is it skipping generations? And then the second thing you would look for is, do you see it predominantly in males? So here, the males are the only ones who have it. The other hint you would have is in a sex-linked inheritance, the only way a female could have it is if her dad for sure has it, and there has to be the possibility that her mom is a carrier. That's the only way a female can get a sex-linked trait. Dad for sure has it, or the mom has a possibility of being a carrier. So here, more males than females are affected. An affected son can have parents who have the normal phenotype for sure, right? Because the son could have he's not going to get it from his dad because his dad's always going to give him the Y chromosome, but the mom could be a carrier, so you wouldn't know she had it, and she could then give it to her sons. For a female to have the characteristic, her father must also have it, and her mother has to be at least a carrier or have the disease. And it can skip generations if it's a recessive trait. All right. So color blindness is one that's often discussed in biology classes. So I know this is annoying, but the allele they always use for color blindness is B, which is annoying because if it's not a dominant trait, it's a recessive trait. So sorry about that, but that's just the way it is. So this would be a normal female, right? She's homozygous. She is not going to have any children that have color blindness. I don't even care if her husband has it because she will always be giving the big B to her children. This is a carrier female. So she could have a daughter who has it if her husband is colorblind, right? Are you with me on that? All right. But if her husband is not colorblind, then her daughter would never have it. Could she have a son that has it? Yes. What do you think, predict wise, how many of this woman's sons would have it? It would be 50-50, right? Because if she has a son, then the Y came from the dad. So 50% of the time she would give the good B and 50% of the time she would give a bad B. If a female is colorblind, all of her sons will be colorblind without a doubt. Okay. If it's a sex linked colorblindness, all of her sons will be colorblind because the dad will be giving the Y and she will be giving this bad X. So all of her sons will for sure. All of her daughters will be carriers. If this woman also married a colorblind man, then all of their kids will be colorblind. Now, a male, okay, this is a normal male. So we don't know anything about his sons, but all of his daughters will not be colorblind for sure. Maybe they're carriers if he marries a colorblind woman, but none of his daughters would be colorblind. This male right here, his daughters will not be colorblind as long as his wife is not a carrier, right? And we don't know anything about his sons because his sons are going to get the Y from him, right? So it just depends on who he married, whether she is colorblind or a carrier. All right, so please do this as a little test of your skill set here. Cross a normal vision man with a woman who is a carrier, a heterozygote, for color blindness, for vision. All right, so do a little Punnett square, get your genotypes and your phenotypes, pause me, and do this cross, giving you time. All right, so hopefully you set it up something like this. So a heterozygote female would be X big B, X little b. She's not colorblind, but she's a carrier. A heterozygote, a normal male would be X big. Males cannot be heterozygote, right? They either have it or they don't have it. They can't be a carrier. A normal male would be X big B, Y. Now, little test right here. What number do you see in each one of these circles? If you are not colorblind, you are seeing the number 45. If you are colorblind, you're seeing a different number. So how would this turn out? Well, like this, right? This would be a normal female, a carrier female, a normal male, and a colorblind male. All right. So Duchenne's muscular dystrophy is another sex-linked trait, as well as colorblindness. All right, hemophilia is a sex-linked trait, and if you've studied your history, then you know in the houses of royalty of European if you know your European history, then you know that hemophilia was running rampant amongst them because if you look right here, 4 children of 9 are shown, but they many of them died of hemophilia. I don't know the number, sorry about that. But because of the way royalty worked, you would keep marrying your cousins, and due to that inbreeding, then hemophilia would kept showing up again and again. All right, these are some more practice problems. You could try them out. Just pause if you need to. And then there's one other one. I didn't put this on your notes, but I want to mention it, and that is that you have something called the Fragile X syndrome, which would be more likely to impact males. This is just another trait besides Duchenne's muscular dystrophy. And that's when you have these repeating bases and of CAG, 6 to 50 copies of that repeat on the X chromosome is perfectly normal. 50 to 230 makes you a carrier. It's a pre-mutation, makes you more likely to have that. That's why they call it genetic anticipation. And then if you have 230 plus repeats, then you have that disease. What's accompanied with it? Mental retardation, hyperactive, autistic, delayed speech. And so that's something that's found on the X chromosome. And if you're a male, you are more likely to be subject to that than if you are a female, because you would have a good copy of that gene, right? So everything we've talked about right there are exceptions to the law of dominance. You're not going to find exceptions to the law of segregation. Why? Because those are always going to turn out poorly, right? When we talked about monosomies and trisomies in humans. The next one we're going to talk about, though, is independent assortment. Oh, put a pause in that. Sorry, I forgot. I gave you a couple practice ones where you could see, do you think here in A, would that pedigree work for a law of dominance? Would it, as a dominant trait, as a recessive trait, or a sex-linked trait? So if you want to, what I would do, I forgot to put this in here, is practice. I'm going to show you the answers in just a minute. If you were given this pedigree and they said, hey, how do you think this trait is inherited? Would it work for a dominant trait? Would it work for a recessive trait? Would it work for a sex-linked trait? And I will do one with you, and then I'll show you all the answers. Okay, so you might want to pause. Pause. Okay, so let's talk about this one. Would this work for a dominant trait? Yes, it would work for a dominant trait, because this person, let's use the letter A, okay? This person could be big A, little a, or big A, I don't know, okay? This one does not have the disease for a dominant trait, so they would be little a, right? So we know then that this girl, their daughter, got a little a from dad, okay, and then got the possibility of a big A from mom, so they have that disease. So that works for a dominant trait. This pedigree is in line with a dominant trait. Is it also in line for a recessive trait? Well, let's do the recessive trait with A's again. So to have the disease, you're little a this time, because it's a recessive trait, not a dominant trait. So this girl's little a. The mom would be little a. The dad could be big A, little a, and he could have given her the little a, right? So yes, that works for a recessive trait. Does it work for a sex-linked trait? Remember what we said about girls. In order for girls to get a recessive sex-linked trait, they need to get a bad X from both parents. So she could definitely get a bad X from her mom, because her mom has the disease. So she could be X big A, right? Or sorry, X little a, and she could have given that X little a to her daughter. But dad doesn't have it. He clearly has a big A because he doesn't have the disease. So he would only give her a big A, X big A. So no, this A will not work for a sex-linked trait. Okay, now try B and C. Now I'm going to show you the answers. Okay, so here are the answers for each of those. So these three different pedigrees, pedigree A, pedigree B, pedigree C. So I've analyzed for you which they would work for. So I'll just do the exception here. So B, it does work for, B, right? B, it works for a recessive trait, right? Because they could be big A, little a, and they could have both given the little a. So it works for a recessive trait. Does it work for a sex-linked trait? Yeah. Dad would give the boy a Y, and mom could give the boy X little a and end up giving him that recessive trait. It does not work for a dominant trait. Do you remember why? Because you cannot mask a dominant trait. One of the parents would have to have it. Okay, and that same thing works out here for C, for all the same reasons that I explained in B. All right, now exceptions to the law of independent assortment. Now we learned with the P's, right? You could get big Y, big R, or big Y, little r, because they were on different chromosomes. Well, you're not going to get independent assortment if all of your traits are located on one homologous pair. So for instance, here, wherever big Z goes, so does big S, big R, and big G, right? And same thing here, little z, little s, little r, little g. They are on the same chromosome. And when they're on the same chromosome, they are referred to as linked genes, right? You're not going to have independent assortment, because if you were doing some sort of dihybrid cross, right, you could think, oh, I could get big Z. If you didn't know they were linked genes, you would expect I could get big Z with big S and I could get big Z with little s. Well, that's not true. Big Z and big S are going together. Even if it was a trihybrid cross, you're going to get big Z with big S and big R. And so the only way you could break that up, do you know? Do you know the only way? Because if they're linked gene, you're going to expect, if you're going to do segregation, right, you're going to get big A and big B, and you're going to get little a and little b. The only way you could mix that up is due to crossing over. Crossing over is the exception to linked genes. If they cross over, they can exchange those alleles. So you could get a little a with a big B and a big A with a little b, right? And so look right here. Most of the time they didn't though, right? 97% of the time they traveled together, and a very small percentage of the time you broke up that linkage. So if you ever do a dihybrid cross and it does not turn out 9-3 to 3-1, if it comes out some other way, then you might consider that it is linked genes. Now, notice here it was 3% recombination. This can be turned into a map unit as to how far apart they are. And let me go back here just to explain that a little bit. Where do you think you're more likely to break up the linkage? Between Z and G, or between Z and S? What do you think is more likely to have a crossing over event? Well, Z and G are much farther apart, so you could cross over anywhere along this chromosome and you would break up the connection between Z and G. But between Z and S, you have a smaller range where you could cross over. So you would probably get fewer recombinants between Z and S and more recombinants between Z and G. And so what you do is, based on the percentage of recombinants you get, that is a map unit, and that helps you to map the genes along that chromosome. Okay, and I have given you all of that in your notes. I've given you some linked genes, some genetic recombination. I even gave you a practice problem that you can do in order to practice that. Just use the notes on that part. All right. So here are 2 other practice problems that you might want to use. And that is it. So hopefully I'll see you in class.